Our first speaker is Esther Shohami. The title of the talk is Traumatic Brain Injury, Short and Long-Term Mechanisms.
Okay, thank you very much. And I'd like to thank the organizers for giving me the opportunity to present our work here and to participate in this exciting meeting. Too bad I didn't have bathing suit last night. Okay. Traumatic brain injury is, of course, very well-known disease. We define it as the mechanical impact that causes the damage to the brain. It's a major public health problem, and I will not go into the demography and the cost and everything. I guess everybody knows. The direct medical cost and the indirect cost is $60 billion in the US 10 years ago, and it's getting higher and higher, I guess. When we look at TBI, it's not a single event that ends when it ends, minutes or seconds or milliseconds, but it's a long-lasting, progressive process which can be defined according to a time scale and events. And what I do today is I talk mainly about the secondary injury, because the primary injury, there's not much we can do about it. It can happen either by motor vehicle accident, some other kind of sports-related accidents, and so on. There's not much we can do about it. And it occurs within milliseconds, and there are axonal tearing and sharing and whatever, but it triggers and it sets in motion many, many events that are responsible for the secondary injury. And that's what is interesting for us. That's what we are studying in our lab in Jerusalem, and that's what everybody else in the field is very well aware of, that there are pathogenics of cell damage, which are mainly defined as excitotoxicity, free radicals, or reactive oxygen species, inflammation, apoptosis. However, in the last decade or so, more and more attention is being paid to the endogenous neuroprotective mechanisms. There are HUJI proteins that are able to help the tissue to recover. There are anti-inflammatory cytokines, there are antioxidants, and there are several mechanisms which may counterbalance the events that are set in motion as the secondary pathogenic mechanism. And in order to study such a complex, and here you see the complexity, such a complex process, we have to have good models, and we have to investigate it along the time course of the events. So what we'll do today is that we look at the secondary mechanisms and how they affect brain damage, the final outcome, and we'll walk through some of these complex events that are mentioned here. Of course, we cannot go into each and every one of them. So what I'll do in my talk is first I'll describe very briefly the experimental model that we have designed and work with, and then I'll speak a little about inflammation, the inflammatory response, the post-traumatic inflammatory response, then on excitotoxicity, and finally on a mechanism that emerges probably as one of the most potent or neuroprotective mechanism that set in motion in parallel to the damage. So the model that we work with is a weight drop closed skull over a free fall device over the skull, which is exposed. We do not perform any craniotomy, any pre-injury surgery, and we can follow up the clinical, motor, cognitive, and depression, and psychological impact, whatever we can do in mice. Several physiological parameters, neurochemical, MRI. It's a good model in mice and rat in mice. Just to show you how the brain looks 4 hours. This is the mouse brain 4 hours after injury. This is the site of injury. Coronal section after Evans blue injection 4 hours after injury. We can see very nicely the focal disruption of the BBB, and of course we did time course to define this process. This model allows us to screen both for drug treatment and for genetic manipulation because it's in mice and there is availability of all the transgenic knockouts, and we make quite extensive use of these tools. When we look at the morphological and histological changes after trauma, we see a week after injury, there is a cavity at the site of the injury. We see dense gliosis. This is the contralateral hemisphere. This is the ipsilateral, the traumatized hemisphere. This is dense gliosis. We see loss of neuron both near the lesion and far away. The hippocampus is not directly affected, but by secondary mechanism, the hippocampus is also affected. CA3 in particular is more vulnerable. In stroke, we know that it's the CA1. Here in trauma, it's the CA3, which is mostly affected, and the effect on the hippocampus may explain some of the cognitive and cognitive deficits that we see. And here are the functional parameters that we look at. This is a clinical score, neurological severity score. It's a composite measure which includes balance, ability to walk on beam, ability to open field behavior, startle reflex, etc. It's a combined parameter, and this is the spontaneous decline in the it's a neurological severity score that with time we see decrease in this score, namely there is some spontaneous recovery but not complete. This is after a month that we still have significant deficit, and these are two different severities of the injury, so we can modify the severity to mild, moderate, according to what we are asking at this time. Here we have a graph of the cognitive memory. I will not go into the detail, but this represents the object recognition test. To those of you who know, the animal that is exposed to two identical objects, and then one is replaced by a new object. Normal animal will spend more time exploring the new object because it remembers the other one. Animal that is impaired, this cognitive memory is impaired, will not spend more time, and here we see the deficits in the severely injured animals. I can explain more about it because afterward, because of lack of time now. And this is the depression that we can measure, the time of floating as a measure of depression. This is a well-known test for antidepressant drugs, and we can see how the animal after a traumatic brain injury suffers of depression, and this is not because the motor deficit. It is, we could dissociate between the motor and the behavior effect. So these are the parameters that we looked at, and here MRI is a good tool that we can do longitudinal follow-up, and the deficit that we see on the MRI is correlated very nicely with the severity of injury. So we can tell that both the severity, this is to really to demonstrate how the model, the validity of the model, because if we look at MRI and it correlates nicely with the neurological score, this is a good verification for us. And here we again see the site of injury, and here is the histology. So all superimposes each on top of the other, and this model has been used in our model for many years, and it's now spread in many other labs around the world use it. And we'll move on to some of the secondary mechanisms that we've been looking at. So we'll start with the inflammation. And as you've seen, one of the early deficits that we can see in our model is the breakdown of the blood-brain barrier. And once the blood-brain barrier is impaired, there is extravasation from the blood of neutrophil, of mononuclear cells, and invasion into the brain parenchyma. Once they are there, they start releasing the mediators and start the inflammatory response. In parallel, also the activated resident brain cells, the activated microglia, and even the neurons respond to the trauma by releasing cytokines, inflammatory cytokines, and thus aggravated the inflammatory response. So the inflammatory, can you see that it's too small or it's okay? Okay, so in the acute phase, as I mentioned, the entry of the circulating neutrophils, monocytes, lymphocytes into the injury site directly affect both neuronal survival and death. So mainly in the early phase, it's mainly death. And the accumulation of the blood-borne immune cells within the parenchyma was measured, was shown both in animal models and in humans. And these activated cells, as I mentioned, release a lot of pro-inflammatory cytokines, reactive oxygen species, complement factors, and all other molecules which are involved in the acute inflammatory response. However, if we wait long enough time, we'll see that the inflammatory response reverses its effect and becomes protective. The brain needs some of the inflammatory response. The microglia over time, first we can identify anti-inflammatory cytokines, or we can find neutralizing binding proteins that neutralize the cytokine that has been released. And they are kind of endogenous mechanism that try to stop the inflammatory response. So the inflammation after brain trauma has dual effects, and I think that the nicest report or the nicest experiment that described it was published long ago in 1999, but it was very clear that, I'll describe this experiment. This is by the group of Sherbel et al. From Philadelphia. What they did, they took a TNF knockout mice and induced trauma in the knockout and in the wild type and followed the recovery, the functional, the composite neuroscore, like I mentioned before, and followed it during about 3 or 4 weeks. And what we see here is that in the early phase, the recovery or the extent of damage in the wild type was greater than in the knockout. Namely, the lack of TNF was good for the mice because the deficit is smaller. Okay? But along the time, see what happens to this line. There is spontaneous recovery. However, the knockouts remain at the same level. Around 1 week, there was a turning point where the wild type were able to recover function and go back to the pre-injury level. And here the knockout mice remained in the same level as they were, so their ability of the knockouts to recover is less than that of the wild type. However, in the beginning, the first couple of days, they seem to be better. So this is exactly the duality that I mean, and here we can really summarize it, and understand what happens, because it's not just a phenomenon. It is very well explained. The major task of the microglia is to remove the cell debris, which is required to attenuate inflammatory process and promote tissue remodeling. However, activated microglia also release neurotoxic substances such as reactive oxygen species, glutamate, and thus exacerbate neural death. Astrocyte reactivity, proliferation, and migration induced after brain trauma seems to impair axonal regrowth. So we have both the yin and yang that we mentioned here, and the presence of these cells around the lesion provides, on one hand, supporting milieu, and on the other hand, toxicity milieu. So it depends exactly at what time after injury we look at the balance between the pro and anti-inflammatory effects, both of the cytokines and the cells of the microglia themselves. So here are just 2 examples from our own study on TNF binding protein, which I got from David Wallach in the Weizmann Institute. He identified this molecule and he gave it to me to test in our model. This is a specific molecule that neutralizes TNF. And in our hands, what we see, this is the water content edema in the brain. So this is the basal level. This is after trauma, increased water content, because we do have edema. And this is after treatment with the binding protein. The level of water accumulation, which is part of the inflammatory response, of course, is reduced after treatment with a binding protein. And here is in the neurological severity score, again, the same score, but it's a different way of expressing it here. But the neurological severity score is better, the functional recovery is better in the presence of the TNF binding protein.